My presentation focuses on the study of the barrier, mostly thermodynamic barrier, between life and non-life, and how it can be overcome during the origin of life process. This is a general picture. In the lower part, main types of prebiotic models are shown. This is RNA world, protein world, lipid world. And in the upper part, this is root of the phylogenetic tree, Thermophilicaceae and bacteria. And this is extant forms of life, progenost or Luca. Let's consider as an example one of the types of prebiotic models suggested by, just this moment, suggested by some Japanese scientists, complex prebiotic model. This is giant vesicle with incorporated DNA. The next step, its evolution, is amplification of DNA due to change in temperature. And the next step is division of giant vesicle along with amplified DNA. Then further process of evolution stops. This is the same process shown in pictures, 10 minutes, and evolution ends. Further, this evolution not moves ahead in the direction to life because this model do not possess two key abilities, two key properties that demonstrate activity of living systems in the environment. First, the ability to boundless expansion and purposeful behavior. So these prebiotic models, as well as other prebiotic models, cannot be transformed to life simply by means of gradual complication of organic matter. There exists a barrier between even the most complex and advanced prebiotic models and the simplest forms of life. There exists a gap between prebiotic models, which are non-living systems, and the simplest living cells that was demonstrated in this picture by the same Japanese scientists. On the left, main types of prebiotic models, on the right, living cells, and between them, mysterious evolution jump or the missing link. And how this missing link could be overcome with prebiotic models on the way to life. Let's consider this gap between non-life and life from the point of view of chemistry and thermodynamics. In the chemical context, chemical evolution, the both and chemical evolution and biological evolution, the both proceed in the same direction with complexification of organic matter. While from the point of thermodynamics, the situation is quite different. We know that all non-living systems exist in the long run with increase in entropy, and prebiotic microsystems as well. While all living systems, especially biosphere, develops in the opposite thermodynamic direction with concentrating of free energy and information. These opposite tendencies are well known. So, in order to overcome the barrier between non-life and life, the prebiotic microsystem must undergo thermodynamic transformation. That means it must transit from the existence in the deficit of free energy or surplus of entropy into the system where contribution of free energy is prevalent over the contribution of entropy. It seems that there exists only way for prebiotic microsystem to overcome this barrier in case it obtains such huge impulse of free energy due to strong change in the environment that compensates its natural tendency to increase in entropy and creates excess of free energy in the microsystem that can be used for further reorganization of the system, increase its surplus free energy and evolution to life. After that, the system must must return to the prebiotic initial prebiotic state with natural increase in entropy due to second law of thermodynamics. But in case changeable conditions in the environment and periodic inputs of energy into the system, it transits into the oscillating regime between initial state of life and non-life. And these oscillating changes of the microsystem, its state, I designate as the intermediate state between life and non-life that is maintained in oscillating mode due to strong and optimal changes in the environment. And from this state, it is possible two ways for further evolution of this system from the intermediate state, either increase in surplus free energy and evolution to life along with biological evolution, or return into the passive non-living state. And these two possible trends of evolution are shown here. This is initial oscillating state of prebiotic microsystems. This is thermodynamic barrier between life and non-life. Sometimes this system obtains huge impulse of free energy, and the system oscillates between life and non-life. In case it accumulates free energy and due to effective response to external changes, it evolves in the direction to life by means of the process of stepwise activation, or the system is unable to concentrate free energy further, and it is degrade with irreversible change, irreversible accumulation of entropy. The same process is shown here, intermediate state between life and non-life, and those possible trends to the simplest microorganism or to any kind of non-living prebiotic microsystem. So there are 4 key points of the concept of thermodynamic inversion. That is a theory. First, starting point, the intermediate state between life and non-life. Second, there exist 2 thermodynamic trends of evolution to active or passive existence. Third, increase in the free energy level occurs through the stepwise activation related with thermodynamic inversion. And the fourth, transition to life was stimulated by effective stress response to external oscillations. Considering microbial world, we can find all 4 points in the anabiotic process of existence of microbial populations. First, this active state, free living form, and below is active existence. Below is passive existence of bacterial cell. This is an anabiotic state. And this is succession of metabolic changes in bacteria when it transits to anabiotic state up to the fully anabiotic state, and the back return to the active state via this succession of changes. And this succession of changes, metabolic changes, when the system returns from the anabiotic state, is opposite the succession of entering it into the anabiotic state. First, when the bacterial population cannot exist further under given conditions, first change is decrease in functional activity of nucleic acid and the enzymatic proteins, then showdown in the movement of lipids and enzymatic proteins in membranes, then dehydration of the cell up to polycrystallization, and the last stage is cytoplasm of a cell acquires a glassy state at critically low residual of amount of free water. This is a final stage. And such passive state, in abiotic state, the system is unable to exit from this state without changes in the environment.
You need to remove that meeting.
So let's compare, on the one hand, 4 key points of the concept of thermodynamic inversion and the concept of theory of anabiosis. The same intermediate state between life and non-life. Second, transition from active to passive state in the theory of thermodynamic inversion, and transitions between active free living existence and the anabiotic state. Third, external stress and response of systems to stress is necessary factor for the origin of life, and in the theory of anabiosis, exit begins also with changes from in the environment, that means from the influence of stress factors. And the fourth, evolution to life from the initial intermediate state proceed through the stepwise activations, like in theory of anabiosis. And this is the following thesis is advanced as advanced for discussion. Since the transition of cells from passive to active state always occur through the same sequence of changes in metabolic processes, it can be assumed that this sequence itself arose in the earliest period of the emergence of primary life forms, and this sequence has been repeating itself even since. This means that the stages of transformation of prebiotic microsystems into primary life forms had been recorded in the stages of exit of a bacterial cell from anabiosis. Regarding the medium of the origin of life, there are three well-known required conditions for the origin of life: aqueous medium, organics, and energy source. And I can add the one more required condition: necessity of multi-level oscillations of physicochemical parameters in highly non-equilibrium medium. Such conditions are absent in ocean, where changes are not strong, and for instance in the ice cover, but they are usual in hydrothermal systems. For instance, these are a picture of Mutnovsky hydrothermal power station in Kamchatka, where vapor used to generate electric power, and these are record of oscillations of pressure and temperature in the wells around this area. We can see at least two levels of fluctuations: irregular macro fluctuations and regular, more or less regular micro oscillations of pressure and temperature. And even the regular micro oscillation with periods of about 20 minutes are very variable, as we can see in these several pictures. In Slovenia, changes in some chemical components also correlate with changes in temperature and pressure. This period about 70 minutes. So hydrothermal systems conditions are very changeable, and they are able to influence prebiotic microsystems stimulate their possible evolution to life. So the most suitable area for the origin of life seems are hydrothermal systems and their discharges on the bottom of ocean or on continents. And coming back to the previous slide, where it was postulated that the stages of transformation of prebiotic microsystems into primary life forms had been recorded in the stages of the exit of a bacterial cell from anabiosis. If accept this thesis, We can use the succession of exit of bacterial cell from anabiosis as a structural basis and succession for the origin of life. And on the basis of this comparison, the 4 stages of the origin of life in the rising hydrothermal fluid have been outlined. First stage, non-living prebiotic microsystems, self-assembly of prebiotic microsystems containing lipid membranes with incorporated polyamino acids as the main components. Second, formation of protocells, intermediate state between non-life and life, appearance of weak energy-giving process of respiration, redox reactions, local watering of the membranes, and acceleration of movement of lipids and membrane proteins. The third, formation of probeins, primary living subcells. This in them genetic apparatus is still absent. A sharp increase in respiratory activity, entry monovalent ions, formation of protein synthesizing apparatus, and acid-base reactions of phosphates, and appearance of primary RNA templates. And the last stage was formation of progenotes, which theoretically reconstructed by Carl Voigt, formation of the growth cell cycle, and the formation of genetic apparatus. On the right side, conditions in geological conditions, first, second, third, and fourth. On the picture, first step, at the depth around 1 kilometer, self-assembly of prebiotic microsystems. Second stage, intermediate state between life and non-life. Higher, then formation of subcells, and the fourth stage is progenotes, and further evolution up to prokaryotic cells. It's supposed that this process was very extended in space and time, because at the earliest Earth, there were a lot of hydrothermal systems. So life, especially its early stages, second and third, could appear not one time. Maybe it was a multi-step formation, and life could originate more than one time in the early life. This is acknowledgements and summary. The origin of life, the transition from non-living prebiotic microsystems to life can be overcome by thermodynamic inversion in oscillatory mode. Second, the sequence of the origin of life process is recorded in the life cycle of a bacterial cell and repeats when it emerges from anabiosis. And third, compatibility of the concept of thermodynamic inversion and theory of anabiosis allow us to distinguish 4 stages of the origin of life on Earth. It begins from self-assembly of microsystems, substantially lipid amino acid composition to formation of genetic apparatus and vegetative cells. Thank you for your attention.
Thank you very much, Vladimir, for a very interesting presentation. Now we have questions. We have time for questions.
Vladimir, what is the relation between your thermodynamic inversion or the concentration of free energy and dissipative structuring in non-equilibrium thermodynamics? Is there some relation there that you can describe? That's what I'm going to be talking about. In fact, the second part, the non-equilibrium thermodynamics. But maybe you could just specify how this comes in relation with your idea or concept.
Yes, I think that non-equilibrium thermodynamics and dissipative structures are the initial component of the microsystem to be transformed into the living state.
So
I use this is a slide which I missed. I use the balance, the total contribution of entropy to total contribution of free energy and information as the most criterion of most important criterion to distinguish non-living and living systems, because in non-living systems, in the long run, entropy increases, while in biological systems, on the contrary, contribution of free energy and information rises. So,
so you would look at as the concentration of free energy as being more important than the dissipation or the production of entropy?
Yes, you are absolutely right. Because dissipative structure is a system which all the time dissipates energy, but nevertheless sustain own existence because of exchange with the environment, with energy, matter, and maybe information in case complex structures. But non-equilibrium conditions or dissipative structures creates initial step for further origin of life. But prevalence of contribution for energy over the contribution of entropy can be reached only by means of thermodynamic inversion, when contribution of free energy becomes higher than contribution of entropy. So availability of dissipative structure is insufficient to create life, but this is a necessary preliminary step for this process.